if we look at the renal system, the renal or the urinary system is not comprised of just strictly the kidneys. Okay, what other structures can we see are making up the renal system? The ureters, bladder, urethra, make up this entire renal system. The major player in the renal system clearly is the kidney. If you're in lab yesterday, you got to look at some kidneys, or today if you're coming, you'll get to look at the primary structure of them. What we're going to study when we fill in our guided notes and look at the actual kidney, it too, like other structures, has a region called the cortex and the medulla. And the cortex is on the outer portion. The medulla is in the middle. And there's another region to the kidney which is called the pelvis, which is this kind of, whatever, pinkish, lighter pink color here. Okay? It's called the pelvis. Between the cortex and the medulla is where that filtration, reabsorption, secretion, all those processes happen. And then the whole point is to try to form filtrate, and then ultimately a small proportion of that filtrate actually becomes what? Urine. Okay? Which would collect in the pelvis and then drain out through the ureter. Can be stored in the structure called the bladder and then excreted from the body and through the urethra. Because the kidney is an example of a reconditioning organ, it has a huge vascular supply. And would you consider the kidney vital or not? It's very much vital, right? How long can you live without it? How long do you think? A few hours, maybe? Okay, so, very much, yeah. You're not going to make it long without medical intervention if you don't have a kidney. If we look at the function of the kidney, the filtering component of the entire kidney are these microscopic structures called what? What are they called? Nephrons. Guesstimated that a million per kidney, so you have a couple million nephrons that are filtering continuously. A high volume of blood is going to them. All of our nephrons have the same basic structure. Some are larger. And remember, these things are three-dimensional structures. And so just like when we study the brain anatomy, something's not always on the left side of the image or the right side or the top or the bottom. You really kind of got to look at the nephron like a maze. And so you kind of got to follow, like it's helpful if you just take your pen or finger and just kind of walk through those convoluted tubes to figure out where you are in order to determine the function of that particular structure. The graded area in the back is illustrating the solute concentration of the interstitial fluid of the kidney. So the light area indicates the interstitial fluid is less concentrated, yet in the medulla, the interstitial fluid becomes more concentrated. So we have a gradient, and we know that gradients allow for things to do what? Move. Okay, this is how our filtrate moves throughout the kidney. This image is taking a nephron that's normally kind of microscopic and twisted upon itself and kind of flattening it out so we can look at the different regions. The molecules that we're going to see include ATP for active transport, salts like sodium, water in the blue, nutrients like glucose is the primary one we really kind of focus on, and waste products, whether they be water soluble or lipid soluble, indicated in the blue and the yellow. So let's go through our regions of the tube. You'll notice that our glomerular capillaries are not shown. Okay, none of the vessels are actually shown in this image. But the tubes originate with which structure? Bowman's capsule. Then we get into the proximal convoluted tubules. You can say proximal tubule or you can say proximal convoluted tubule. The convoluted is in there because it's all twisted. To do what? Why do we see this thing twisted? Things twist and fold to greater surface area, right? So we're just packing it in there. So, Bowman's capsule, proximal convoluted tubule, then the filtrate would flow through the descending limb of loop of Henle. And then turn back and then hit the ascending limb of the loop of Henle. Then we'll hit the distal convoluted tubule, then to the collecting duct. Along the way, we're going to see a procedure. We're going to see filtration, reabsorption, secretion. Hands down, filtration has to happen first, but reabsorption will happen in high proportion. The secretion may happen as well initially, but we're going to see first a whole bunch of reabsorption and then finish out with secretion. So let's kind of look at it in motion, and it'll help this last chapter go a little bit easier. Okay. So the first process of urine formation is called filtration. Remember, these are capillaries, but they're special. They have really huge pressure, and we'll identify, if not today, better tomorrow. So high pressure, so filtration only, but we don't want these products to be, we don't want them to be reabsorbed back to the glomerular capillaries. And so we'll talk about the mechanics between which arterioles. Which one is this? You think? Afferent and the efferent. Okay, so it's going to have something to do with how those afferent and efferent arterioles work to make sure that filtrate is formed, okay, but not stuff being reabsorbed. Just like any other capillary, what is not filtered out? And the point of kind of today's lab, what's not filtered out your capillaries?
Protein.
Big proteins, nor red blood cells. Anything else that's able to fit through the pores or is transported or whatever, getting pushed out. And whether it's glucose, those hormones you just spend all the time trying to make, they're getting pushed out. But we can reclaim them through this process we can see here called what? What? That's not actually exocytosis, so the whole process of moving filtrate back into the blood, as we said, is called reabsorption. Something, I don't know of any vesiculation that may happen, but I'm not aware of any that would happen in the tubules. We usually see active transporters, primary and secondary, and diffusion and osmosis. So we got glucose carriers, aquaporins, all kinds of transporters chock full here in that proximal convoluted tubule. So can you see the reabsorption of, it says here, nutrients, okay, the green things, actively reabsorbing. Okay. Anything else that's not reabsorbed stays in the tube. Okay, and we're calling it what now? Here it's called filtrate. Here it's called, in a capillary it's called plasma. If it's in the capillary, it's plasma. If it's in the tube, it's filtrate. If it's between the two, we would call it interstitial fluid. Okay, so the new term to us is filtrate, being trapped in the tubes. So we can see some initial reabsorption after filtration along that proximal convoluted tubule. The filtrate then will reach an area called the descending limb of the loop of Henle. And notice which molecules are more selectively reabsorbed here. What is being more selectively reabsorbed here? Water. This can be hormonally influenced by which hormone we've been talking about for about 6 weeks. ADH is right, antidiuretic hormone. Directly promotes water reabsorption at the kidney tubules. So antidiuretic hormone can target the descending limb of the loop of Henle for the reabsorption of water. Okay. Aldosterone can influence the kidney tubules, yet at the which limb? Ascending limb. If you remember, aldosterone selectively promotes sodium reabsorption, so it's on the ascending limb, which aldosterone can target at the kidney. As filtrate travels from the ascending limb of the loop of Henle, then it'll get over to, we'll start, this is the peak of the ascending limb where we still see some more reabsorption of our salts, if you will. And then we'll get over to the distal convoluted tubule, where there's still some reabsorption going on, but we also see a greater proportion of what? Secretion. So if you remember, for example, aldosterone promoted sodium reabsorption, but what? Secretion of potassium. Very good. So here would be an example of potassium being secreted back from the interstitial fluid into the filtrate. Things that are waste products like urea that are lipid soluble would just diffuse from the interstitial fluid back into the tubes. After the distal convoluted tubule, the filtrate would flow into an area called the collecting duct. And at the collecting duct, the filtrate is fairly well modified. There may be some more water reabsorption, maybe a little bit of salt, but very little modification. Not the magnitude of reabsorption or secretion like we saw at the rest of the tubes, but it can be modified a little bit. By the time the filtrate reaches the end of the collecting duct, it'll enter into the pelvis, and then we can call it the what? Then you can call it urine. You don't call it urine prior to the end of the collecting duct. Why? Why wouldn't I want to call, say this right here, urine? It's still being, not filtered, but still being secreted. Substances are still being secreted, added to our reabsorbed product. So we're still modifying it. So we still have a chance to reclaim these substances back into the blood. And so we don't want to call it urine at that point. But once it's done being messed with or modified, as long as the reabsorption secretion is not happening anymore, then you can call it urine. And that is crash course in urine formation and how it happens, the three primary phases. First, we said the first phase is filtration. Then the filtrate can be reabsorbed, as well as interstitial fluid can secrete back into the filtrate. All right, so let's just go ahead and pass your handout number 7 to the middle of the room, to the middle of the end of your table towards the center. And I'll pick those up in just a moment. And then we'll start our chapter 18-19 guided notes. Again, just like throughout the rest of the semester, you're going to return to those reading pages. It's outlined in your syllabus. Chapter 18, it looks like about 25 pages and about 30 pages from chapter 19. So not too much compared to the collective chapters. Okay. We are familiar with the kidney. Sorry. Stop this. It's moving all at once, I guess. The job of the kidneys is to filter blood, and they can filter about 200 liters of blood per day. Do you have 200 liters of blood in your body? You have about how many liters? 4 to 6, right? So this tells us that the kidney is a reconditioning organ. It receives not only just adequate blood flow, but excessive blood flow. And the point is to get rid of excessive products, true waste products, toxins, whatever your body may be exposed. It's a way to get rid of unneeded or excessive substances. And thusly, when it does this, it influences the blood composition, both the water and the solute. Okay, so the solvent and the solute composition of your blood, to make sure that we have adequate set point balance between water, solutes, hormones, etc. In our body. The renal system will also make sure our blood pH is adequate. It'll pick up where the respiratory system leaves off. Okay, so it's a more finite solution to blood pH, but it does make sure acid and base balance is adequate in the blood. If we look at the organs in the renal system, we identified in the previous animation that it's not just the kidneys. We have also the ureters, the bladder, and the urethra are components of the renal system. We also studied the structure of, those structures are summarized for you in figure 18.1 in your textbook, looking at the organs there. And if we were to look at the structure of the kidney, it has those three areas. What were the three areas? We had the cortex, renal cortex, renal medulla, and the renal pelvis. Very good. It was a good job including the prefix renal. Everything, other structures have a cortex and a medulla, so we're looking at the renal. The cortex, the renal cortex, is the outermost region. The medulla is the area described in your handout or guided notes as cone-shaped. They're kind of triangular-shaped areas. We'll see those in our kidney dissections today, or you did yesterday. Between the cortex and the medulla, we will find tubes and our capillaries in there as well. And the pelvis is this kind of connective tissue-y structure. It looks like a funnel. I mean, it's described as a funnel. It looks like a funnel that you would use to put your oil in your car in. Okay, so it kind of looks like that, but it's connective tissue. And that's what it's doing. It's funneling now urine right out of the kidney down to the ureter. The image from your textbook, maybe figure 18.2. I don't have it shown for some reason. But if we look, where I'm pointing here, that thin layer is called the cortex. In this image, the kind of darker pink areas are called the medulla. And then what's colored yellow is called the pelvis. That's where the urine will collect. The kidney doesn't have this yellow color. It's just colored like this for you to see in the book, as well as we know that our blood is not blue, right? So the same type of coloration. But the kidney is kind of a dark color, kind of a not quite purple, maroon kind of colored, slightly, right? Not real apparent, but kind of a darker colored structure. And if we look closer between the cortex and the medulla, what's making that up is the tubes that we've already seen in the image, and that dense vasculature structure with all that capillaries and arterioles and venules, etc., forming our filtrate. And you'll notice in the medulla, primarily our loops extend down the medulla, as well as our collecting ducts extend right throughout and dump into the collecting duct. This is the image which we used for handout number 7 that you just turned in to identify the different regions between the capsule, proximal convoluted tubule, descending limb of Luca Henle, ascending limb, and our distal convoluted tubule, and the collecting duct. These structures, I believe, are already labeled for you in your guided notes, but I'd like for you to kind of practice and try to identify the regions. We know that the working bit of the kidney is the nephron, and we said the nephron as vascular or our blood supply. It also has tubes that transport what? Tubes that transport filtrate. Okay. What color are the tubes in the image? Yellow. The vessels are blue and red and purple. Okay. So let's go through, and it's labeled for you, so let's just add some directionality. Let's review the directionality of blood flow through our vessels and the directionality of the filtrate through our tubes. Which do you guys want to do first, the blood or the filtrate? Let's go to the filtrate. Where is the filtrate formed? Where is it first formed? Where do we start the filtrate journey? Bowman's capsule, which are actually surrounding our glomerular capillaries. And so this little structure in here, the Bowman's capsule, you can see how it encapsulates and captures filtrate. So filtrate is being pushed from the capillaries towards the tube. So if you can, I don't know how this is going to work in your handout, but try to squeeze a little arrow in there to illustrate the filtrate moving away from the capillaries. Filtrate would then flow, push through this area called the proximal convoluted tubule. Maybe you want to draw an arrow here to illustrate it's still going away. Falling, maybe you want to add another one here, still moving away. moving down. And then now it's moving which way here? Descending. So here, draw a little arrow down, down. And go ahead and label that the descending limb of the loop of Henle. Filtrate would move in a downward fashion through the descending limb of the loop of Henle, hit the loop, and then move which way? We describe that as ascending, and it is moving up. So go ahead and put an arrow up. Up. And it continues through. Okay, we're going to draw another arrow up till it reaches an area. This is called the distal convoluted tubule. And then it will drain into the collecting duct. It's moving in a descending fashion to the collecting duct. What questions do you have about the directionality of the filtrate through the tubes? Let's go back and see how it was first formed. So we've got to look at our blood vessels. Bringing blood to any organ is an artery, and the arteries branch off to arterioles. But we learned that the nephron is different. Then it has some directionality, an afferent and an efferent arteriole. Which arteriole brings blood to the nephron? Afferent. And this one's labeled for us. So go ahead and just draw an arrow for your blood moving towards the glomerulus. And that's where we started earlier. We said it forms filtrate. Any blood that is not filtered then enters into which vessel? Efferent arteriole. So if you can, squeeze a little arrow to point this away-like fashion or movement away from the what? Where are we moving away from? Away from the Bowman's capsule, away from the glomerulus. Blood from the efferent arteriole then drains into, what are these purple things? They're called paratubular capillaries because they happen to be where? Around the tubes. So it's just going to mass enter the paratubular capillaries, which is going to work with them for reabsorption and secretion. After the paratubular capillaries, then we would enter into our familiar venule, and then eventually into the renal vein. What questions do you have about the difference in the flow of filtrate through the tubes versus the flow of blood plasma through your blood vessels at the nephron? All right, let's just conclude this introduction to the nephron. We know that the filtering part is at the glomerular capillaries. And it's surrounded by the Bowman's capsule. You could call it the glomerular capsule or the Bowman's capsule. It surrounds the glomerulus, and its purpose is to not only surround it, but to collect what? To collect filtrate. The glomerulus makes the filtrate, the capsule captures the filtrate. And if we look at, we'll just kind of conclude on this first page real quick. We do have two capillary beds. We know we have the glomerular capillaries, and we have also now the paratubular capillaries as well. We know the glomerulus is unique, and that receives blood via an afferent arteriole and is drained by, did I misspeak? It's fed by an afferent arteriole, yet drained by an efferent arteriole. We'll conclude with this introduction, and your job is to read chapter 18 and 19 reading pages over the evening. We're going to also conclude this semester with that renin-angiotensin mechanism pathway. So we're going to go back to this and that and that and that kind of pathway. So come prepared for our last day of class tomorrow.